that actually at the previous talk was there, because now that we've discussed what neuroscientists should do in order to achieve whole brain emulation, I will try to show more or less what they have been doing, especially in the past year, and how just how fast neuroscience is changing in order to achieve exactly this. Then I will try to show that there are some sort of pressures within neuroscience in terms of both publishing and funding to achieve whole brain emulation. And then, of course, finally to explain how this is relevant for AGI, because that's the point of the talk. So we're starting with our common friend here. I must warn you, this talk will be very physiological. At some point I will mention the action potential. At any point, please tell me and I will go into more details. So in a previous paper called Available Tools for Brain Emulation, I tried to put down all of the methods that are currently in use in neuroscience. So one main problem is that the closer we are to the human brain, the harder it is for us to become invasive due to ethical constraints. So in humans, our main tools are FHI and AGI, which are good tools in principle. It's just that what functional magnetic resonance imaging does is to measure changes in blood flow, while the electroencephalogram just measures changes in electrical potential on top of a very strong isolator, which is the human skull. Sorry, the strong tools are mostly used in lower primates and mammals. So here we can use microscopy, single cell electrophysiology, and genetics analysis. Right. The best models for doing genetics are the lower animals, because they provide a simpler system, and then we can more easily verify the function within these systems. So today I will be talking about this paper. It was written in Science in 2011, and it is written by Winfried Denk and Jeff Ligman. So Winfried Denk invented two-photon microscopy, and Jeff Ligman invented the... Sorry, am I being too... Can you hear me?
No, not very well.
I don't know where the microphone is.
So should I get closer to the microphone?
Yes.
There's another one, it's a roving one, I don't know where it is.
Like this?
Yes.
Yeah, that's it.
Where's the roving microphone?
This one here?
It's a bit of a strange
Yeah.
picture.
This one actually might be better.
Okay, or that, yeah. I'll try again, can you hear me now?
Yes.
Okay, so what this paper does is to basically put down the main problems in neuroscience. So in 2011, they noticed that there are 5 big milestones in neuroscience, and they basically say where neuroscience was at that particular time. Now we are in 2012. Of course, the main problem in neuroscience and for whole brain emulation in general was bridging structure and function, which in the brain, as opposed to any other organ in the human body, has been a very, very difficult math. So problem number 1. There are about 65 billion neurons in the human brain. Each neuron has around 7,000 synaptic connections. If we multiply these numbers, we get more than the number of atoms in the known universe, which is quite a bit of complexity. Now, within this, there are approximately 200 cell types that have been identified with their structure and their functions mapped. This is very little, and at the moment, the field doesn't have a very good idea of how many other cell types there would be and what their function is at the higher level. The second problem that they point out is connecting electrical and chemical activity. So the connection between chemistry and physics has been a philosophical problem for a long time. The good news in the brain is that this is actually not so complicated. So there are 3 main ions that interest us in the human brain, and these ions are sodium, potassium, and chloride. Okay, now I'm curious because I wanted to go into the action potential. Is anyone bothered by this slide in particular, or should I go through it? I see something. Right, so to explain what you see here in the picture on the left, my right to the left, these lines that you see going into these neurons, they are electrodes, and what they're measuring is changes in membrane voltage. The other traces that you see on your right the colorful ones, they are calcium transient. So what you will see in these dendrites here is changes in the concentrations of calcium, which appear as these peaks here. And these two things are actually hard to correlate, especially because of spatial summation. In neurons, because they are such complex circuits, like I said, there are more than 7,000 synaptic connections on each of these neurons. We find this phenomenon called nonlinear summation. This basically just means that the input is not the same as the output, but you might imagine, knowing what complexity is, how that might work within a circuit. So what happens at the level of the membrane? So when the signals from all of these dendrites reach the soma, where you see the electrode here, what is being recorded is what you see here. So the resting membrane potential is around minus 65 millivolts. When the signals that reach the soma all together, if they sum as a strong enough input, which would take it over minus 55 millivolts, so making it more positive, then an all or none event will begin, which is very similar to that of explosion. And the membrane will become more and more positive up to, say, plus 40 millivolts. So this principle is very simple. It just means that when the membrane becomes more positive, more sodium channels open, which causes more sodium to come in, which causes more sodium channels to open, and so on and so forth up to plus 40. At plus 40 millivolts, it will go down because the potassium will keep on leaving the cell, and it will cause the membrane to return to its resting value. So that's pretty much what happens with the neuron. That is what is being recorded most of the time in different forms. So this is what happens when we look at the actual living brain, when the brain does stuff. Right, problem number 3. So neurons extend to very vast volumes. Although the soma of the neuron, the nucleus that you see here, is around 20 micrometers, a whole neuron, as you can see in this Causal sketch, it can go from one hemisphere to another, so it can go be as long as 1 meter, actually. Now, looking at this at a very high scale can be very difficult. So imagine we have to look at the inputs which come in the dendritic spine. These are at nanometer level. Mapping a whole neuron at the nanometer level in vivo can be very difficult. And this is why they point out this problem. Problem number 4 is actually not a big problem anymore because, as I will point out later, so the detailed structure, they say, cannot be resolved by traditional light microscopy. This is very true. It was true 20 years ago, but not anymore, since super resolution microscopy is now in use, and well, most self-respecting labs have them nowadays and use them. And the last problem. So the need for dense or saturated reconstruction. So what they are saying, again, we're talking about nanometer scale events, electrical and chemical events. One has to reconstruct this. So it is assumed that the human brain is functionally organized within columns. So this would be a cortical column, for example. So one would have to map an entire column like this at the nanometer scale, and we would need to have actually run a movie of this. It is not sufficient to have, say, a 3D image, because this would give us no clue about the function of that thing and how it changes due to the environment. Right, so now we know the problems. And what I wanted to show you is the approximate growth of neuroscience over the years. So on the x-axis, you have the year. So it starts from 1979 up to 2011. And then on the y-axis, you have the number of publications. And you see here, it goes from 50,000 publications in 1979 up to 250,000 in 2011. So this is actually a very, very fast growth. This is the point of this talk, in a way that, although the field is extremely young, it is also growing extremely fast due to high interest. This attracts funding and therefore causes a lot of publications and a lot of data to be provided and modeled. And one big problem that was pointed out before, so one has to connect structure and function. In order to do that, one needs to look at these structures in the living animal. For this reason, there had to be much more experiments in the living animals, so in vivo. And this is again a graph which shows the growth of in vivo publications. So from 2092, it goes to 12,000 in 2012. And here you can actually see how steep the slope is from 2009, 2010 up to 2011. Right, so now that we had these things in mind, now that we know just how fast the domain is evolving, we can go back to each problem. I cannot, of course, go into a lot of... Is there a problem?
The system just does that, don't worry about it.
Never mind. Right, so now that we know just how fast these problems might be resolved, I'm going to refer to the problems mentioned in 2011 with the information that we have now in 2012. Right, so the problem again, the immense diversity of cell types, we had 64 billion neurons, 200 types of cells documented approximately. When I say that new types are currently being mapped, I mean take 200 and think exponential. So the number of these projects is far larger than what is currently being published, and that is indeed a problem. The speed of publication is also limiting our access to this kind of information, but there are many, many repositories and databases. At the same time, new methods are created for making this process more efficient, so for mapping these kinds of cells. Again, I'm happy to mention any of these things later on in the Q&A session. So the second problem was coupling electrical and chemical activity. So we had the problem of nonlinear summation neurons. This is hard to explain in vivo, and especially at the large scale. So nowadays the solution would be that many calcium indicators have been developed. So calcium indicators can basically show what's happening with that action potential with the sodium coming in. And it's actually showing this as a direct function of it. Also voltage imaging can be used, so this is a direct change in voltage that is being measured. As well as this new project with large-scale electrical recordings with nanoprobes. So if I read the paper correctly, the idea was that these silicon nanoprobes would attach to every neuron, and then you could record from hundreds and hundreds of neurons at the same time. Right, problem number 3. So they extended over vast volumes. The new methods for this confocal microscopy is now very new. Super resolution microscopy and two-photon microscopy with their variants are actually very useful for tracing entire neurons. Their newest versions actually allow for real-time tracing within the living animal. So while one image is on a neuron, they can actually see live where exactly they are in that particular neuron. Problem number 4, with the detailed structure that cannot be resolved by traditional light microscopy. Again, the same thing as in the previous slide, there are new methods which are made specifically for this. So in vivo super resolution is now standardized, not really a problem anymore. Finally, need for denser saturated reconstruction. So, as I was saying, one would need large-scale movies correlating structure and function in cortical columns. There are many projects running in the Connectome consortium. New methods for tracking connectivity have been found, such as the rabies virus. And most importantly, the scientific community is reorganizing for accommodating such tedious projects. So in fact, tracing entire neurons would mean hundreds and hundreds of hours of work, and it would require actually the work of hundreds of people. So the domain has to reorganize in order to accommodate such projects with so many people. Finally, the reason why I gave this talk. So the implications for AGI. Now, neuroscientific research is inevitably gathering connectome data. Everything that I've referred to before can be called connectome data. This data is currently being acquired and modeled by neuroscientists with the end goal of achieving a complete connectome. This end goal was never connected to AGI in particular. It is just something that is automatically coming out of neuroscience. One would imagine that this would be the end of neuroscience, right? So what other final goal would it have in understanding the whole brain at a very large scale? Now, once neuroscience provides a connectome, this should be easily rebuilt in silico once all the information is made available, and this would constitute a whole brain emulation, and why not an AGI? Now, you might ask me at which scale one would need to have this connectome in order to achieve this brain emulation. My answer would probably be atomic scale, because without knowing the exact structure of the molecules that allow for plasticity in the brain, which is a very special phenomenon, but it's the only thing that allows us to learn, then the system will not be adaptive to the environment, at least not in the same way that the human brain is. So with this, I would like to thank you, and I'm happy to hear questions.
Thank you. Questions?
They went up almost simultaneously.
Just a comment on your very last slide about achieving these aims would be the end of neuroscience. Now, of course, I'm sure you don't mean that seriously, but if we did achieve those aims, of course, the really great challenge would still remain of understanding the thing that we'd reconstructed and deriving the principles that explained what was really going on. And what's really lacking in these 200,000 papers that are coming out now are a really deep set of principles analogous to Darwin's theory of evolution, which underpins the whole thing. And we're really, I think, lacking that in the brain sciences. And from the perspective of AGI, it seems to me that if anybody ever had the ambition of whole brain emulation followed by radical enhancement of some sort, then in order to achieve radical enhancement, you need more than just reconstruction. You need understanding in order to re-engineer things and so on. So those principles, extracting those principles, to my mind, seem like a really the really great aim of neuroscience. To which, of course, projects like this are absolutely vital. But I don't think we'd have got there even if we succeeded with this kind of project.
Actually, there was a
Use this
hand
microphone?
there a little bit earlier.
No, it's okay. I can use this one. Right. So I'm wondering now what you might mean by understanding in neurophysiology, because to a large extent, okay, we can say that we understand some principles, but what we mostly do, it seems to me, is just that we're observing things, we're counting them, and then we are trying to find some commonalities or patterns between them. But I think the moment where you can construct a model which does exactly the same thing, at least to your understanding, I don't think that there is much more understanding to be done after this. I'm not sure, but yeah. In that sense, AGI would also support neuroscience, right? Maybe it provides the understanding we don't have.
Thank you very much for what I thought was a really excellent review. The point I wanted to make was that the connectome is not genetically determined, but the different kinds of ion channels are genetically determined. And there's quite a wide variety of these. There are hundreds of ion channels. Christoph talks about the soup of ion channels. And there do appear to be differences between different people's and different animals' brains that are based on those. So that's the first thing I wanted to say. The second thing I wanted to say is that rather than trying to build it atomically, for which people like Fujitsu and IBM would probably thank you because you would need most enormous computers. Perhaps an alternative might be to understand its genetic basis and to, in some sense, to grow it.
Yeah, so I completely agree. The genetics is obviously vital for understanding the structure of ion channels. But this is kind of related to the previous question. I think we need an atomic scale structure of these proteins in order to understand their function in the system. And of course, once we have the structure of these ion channels, we would definitely need to see how the genes produce them, right? And why their distribution is different in different humans, and whether that has any relevance, actually. Maybe it's just randomness and it works in the same way. Yeah.
I'm Alan King, and my question is, if my learning something involved extending axons in a connectivity kind of way, as opposed to just synapse strengths, do we have the technology to observe that right now?
Yes, so the dye is called GCAM6. It recently came out. And it's used specifically for imaging axons, synaptic boutons. So you can do calcium imaging with them and directly verify the structure, maybe couple of them. Whoa.
And do they show axon growth?
Yes, one can follow this. So in a quantic experiment where a quantic window is implanted, yes, you can see exactly. but I shouldn't be telling you this because it's not published yet.
My question goes in the same direction as Leslie's question. What makes you think that it's easier to simulate 10 to the power of 27 atoms or something than to determine what a neuron contributes to information processing and what maybe a column contributes to information processing? What makes it so inscrutable?
So, yeah, I think the idea of checking what one thing does to contribute to the whole system can be a very good idea. But I think we might actually be missing the point. It seems more and more obvious that the higher level functions are supported by complex processes. And therefore one would need to understand how networks, entire networks interact.
Okay, I have 3 other questions at least here. And I know you put your hand up a little bit earlier as well, so I think,
I'm somewhat concerned about the possibility that we might need atomic precision, because that would probably mean that we're never going to get any brain emulation. However, I'm more optimistic. The real question that I think it would be very worthwhile to pursue is, does there exist scale separation at some size scale in the brain? For example, in statistical mechanics, there is a very strong scale separation between the microdynamics of air molecules and the pressure, temperature, and other macro variables we observe. And since very decoupled from each other, you can average things very nicely, and you don't need to think about what molecules are in there. Now, if the same thing would be true in the brain, then you just need to find that separation there. Conversely, if it turns out that there is no such neat scale, then we have a very good argument that brain emulation is probably never going to be feasible. It might produce a lot of good neuroscience, this kind of research, but we're not going to be uploading ourselves in any way. So what forms of research do you think this technology could help us do to settle the scale separation issue?
Yes, thank you very much for the question. So, in terms of scale separation, we should always keep in mind that there are always causal connections between the different scales, right? So, it is true that we are looking at different scales with different tools, but that does not eliminate the fact that, for example, the distribution of ion channels is what generates plasticity and learning. So when we look at learning, we actually need to keep in mind the ion channels and the genes and the molecular structure and all of this stuff. I said atomic scale for whole brain emulation because I think the idea with whole brain emulation, as I understand it, is that you have something as close to the biological brain as possible. If the idea would be to keep as much as the functions as possible, like in AGI, or to pre-specify some functions that one would like to emulate, then the atomic scale is really not a problem for us. We could, of course, just stop at the circuit level and then just, let's see how many other neuron cell types we have, and then we can just rebuild the whole circuit. Yeah.
let's have one more question, and I think there was one here in this corner. Yeah.
Hi there, great talk. I guess there's coming at this from a non-neuroscientist or a lay perspective, there's a lot of people who are interested in the general idea of what is the model of intelligence in the brain, and there's been some books like Jeff Hawkins and also Ray Kurzweil's new book. He spends quite a bit of time discussing a model of how the brain might work with these cortical columns, and you've got this hierarchical temporal memory models and all this type of stuff. So is there a discipline within neuroscience which tries to kind of integrate these conceptual models for general intelligence and the actual, and the neuroscience? Is there an actual discipline that exists where they're trying to kind of bridge these two areas, or is it just kind of like individual researchers or individual institutions that are kind of like not really connected together?
Thank you. Yeah, this is a difficult question. The way I think of intelligence is that it might be related to some other social construct. So I think that we tend to define intelligence in connection to other people. Like if X can do something I cannot do, then I say X is intelligent, right? So this is in a way, to me, more connected to machine learning or psychology or some other things. I also think that the brain does not care in particular about our categories. It just does whatever it does in the most efficient way possible. So yeah, I think intelligence is a very complex concept, and it's very hard to combine it with the brain, which is a system which doesn't care about our categories. That's my conclusion. I hope I answered your question.
Not quite. I'm just trying to figure out, is there any organizing principle or anybody who's trying to sort out that we have these high-level models that people are speculating about how intelligence works now, and these ideas of hierarchical memory models and cortical columns and pattern recognizers and these types of things. So it's instead of looking at the brain as a whole, but more kind of focusing on that conceptual model. Is there an area within neuroscience which is specifically just trying to work on this from a theoretical down to the...
Okay, so I also think that the neuroscience is sort of lacking a rigorous understanding of very high-level functions, and this is because these we mostly find in humans, and humans are not very accessible right now.
There are, of course, areas in neuroscience that try to do that, like cognitive neuroscience, for example, which sort of connects to cognitive science and then everything else. So there are definitely fields that aim at doing this. Although then again, they have to sacrifice a lot in terms of what they know about, or take into account about the neurophysiology. So, and I'm afraid we really have to move on because we are already going to be extending slightly into the coffee break the way things are going. Thank you very much. That was an incredibly